All right, picking up where we left off, here we have, again, glucose, two glucose molecules coming together to form maltose. Here we have a hydroxyl chemical group here. Here's another hydroxyl. One of the oxygens and both hydrogens are going to be removed as a single water molecule. That, again, dehydration reaction. This forms this linkage of a carbon, oxygen, carbon, which we call a glycosidic linkage. Okay, we refer to this specifically as a 1,4 glycosidic linkage because if you label the carbons 1, 2, 3, 4, 5, and 6, this is a linkage between 1 and 1, 2, 3, 4, 5, 6, so a 1 to 4 linkage. Here we have glucose coming together with fructose. This again is forming something called sucrose, which is also known as table sugar. Again, hydroxyl reacting with a hydroxyl, creating a carbon-oxygen-carbon, and in this case it is a 1 to 2 linkage. Okay, just so happens that this sugar is labeled slightly differently, so this is actually carbon 2. And this is based on where the carbonyl is in fructose. Okay. The numbering is always based upon how close that carbonyl group is. So here in carbon 1, it is where the carbonyl group is. Here, the carbonyl group is present here, so it is carbon 2 because we have 1, 2, 3, 4, 5, 6. I'm sorry, 1, 2, 3, 4, 5, 6. Yes, this is 6 carbon sugar. All right, so we can extend this out and beyond disaccharides and start looking at polysaccharides. Okay. Polysaccharides are just longer and longer chains of sugars. These polysaccharides, particularly these long chains, typically have a storage function in storing that sugar material, typically for fuel usage or as a structural role. Okay? How these molecules come together and their larger overall structure is going to give this architectural feature and consequently its function. Remember, structure is function. These structures are determined by how these monomers are linked together. Okay, so let's take 3 examples of what are actually polymers simply of glucose. Okay. So here is glucose coming together. This is coming together in a 1 to 4 linkage. And we see this in multiple locations, right? So we see this in starch, which comes from plants. We see this sugar chain coming from something called glycogen, which is our own cellular sugar storage material. We also see something very similar in cellulose, okay? But its overall structure is slightly different. So let's go into that. Okay. So here we have starch and glycogen. They actually have a very similar structure. Okay. So we have these glucose and 1 to 4 linkages. When it's unbranched, we call that amylose. When it starts to become branched, we call it amylopectin. Okay. When that sugar is sourced from us, as opposed to potatoes, we call it glycogen. Okay? Structurally, there is somewhat of a difference, not in the actual chain zoomed in itself, but glycogen tends to be much more highly branched than we see starch. Okay? But the fact that they look more or less similar has a functional significance. Here, if we look at the cell walls of plants, which is made up of these things called cellulose, here we have cellulose larger microfibrils, we can break those microfibrils down into these long individual cellulose molecule chains. Okay, these are unbranched, and notice that they have somewhat of a different arrangement. Notice how these oxygens are all appearing on the same side. Here we have all of the oxygens appearing on the same side. Here, those oxygens appear to flip-flop. Okay, there's a reason for that. The reason for that is when we build these molecules, the enzymes that do it are looking for a particular form of glucose. And remember, when we form a glucose ring, we have two options. We can form it in an alpha configuration with the hydroxyl below the ring, or in a beta configuration with the hydroxyl above the ring. Okay? It just so happens that we and potatoes are looking for this. Plants producing cellulose for their cell walls are looking for this, right? They look different to the enzymes. Consequently, when we look at these chains, it has a structural significance. Here we have starch, so it's a 1-to-4 linkage, or we could call this an alpha 1-to-4 linkage. This leads to this configuration where we have these hydroxyls, or these sugar rings, essentially all facing the same direction. Okay? Here, because we are linking a beta linkage with it above the ring to a carbon where it is in a different position, we end up with this flip-flopped structure, right? So here we have a hydroxyl, we come up, here's the glycosidic linkage, it is kind of arrow pointing up. This one is now arrow pointing down, and this hydroxyl is on the opposite side. And you can see this reverses back and forth, back and forth, back and forth. Okay? When the enzymes are producing these molecules, it is looking for particular formations. So here, the enzyme making cellulose is looking for beta glucose. When potatoes are making starch, they are looking for alpha glucose. Okay? The same is true for breaking those down. So the enzymes that break down or digest starch by hydrolyzing these alpha linkages are expecting to see this structure. Okay? If it sees this, it doesn't know what to do with it. So consequently, the enzymes that can break down starch cannot break down cellulose. The fact that we use glycogen, which is a very similarly related molecule of alpha linkages, we have an enzyme that can digest this, so it's very easy for us to digest potatoes, but we cannot digest this cellulose material. This, in fact, passes straight through our body and is referred to as insoluble fiber. Okay? There are some microbes that have the ability to digest cellulose, and they are actually responsible for the ability of herbivores to utilize cellulose as a material. They essentially form a symbiotic relationship. They have a separate fermentation chamber in their digestive tract that houses these microbes. The microbes are given a nice warm place to live and constantly be given new food. The microbes turn around and give some of that food to the herbivores. Okay. Now, this structural difference of alpha versus beta linkage can also have other repercussions. Okay, so starch and glycogen, if you look at it, forms these kind of helical structures. So here we have a helical type structure. It branches here, but then it goes back into a helical. We see the same thing with glycogen. Okay? In cellulose, the fact that we have this flip-flopping nature adds the ability to form hydrogen bonds between the strands. Okay? This is actually what leads to the ability of cellulose to come together and form these larger fibrils that can then form these plant fibers, which provide the structure. Think of the woody material in trees, right? This can be a very strong structure when added together, okay? So for here, both of these two circumstances, these larger sugar molecules are being used as a storage material. The whole idea is when you run out of food that you're digesting, you can start breaking down these sugars and use them instead. Okay, for the potatoes, in the early springtime, before it's produced leaves for that season, it utilizes the energy stored in those tubers to start growing new shoots until you can develop new leaves to start gaining energy from the sun. Okay? For us, when we run out of food, we start digesting this. This cellulose is acting as a structural element. This is not unique to plants, right? So if we look at insects, insects have a very similar polymer made up of this material called chitin, which is essentially glucose with this little added group onto it. But it is put together in beta conformations and long chains, which has the ability to form hydrogen bonds between chains, and it can produce this hard exoskeleton of insects. Okay, this chitin material can also provide support in the cell walls of some fungi. We can utilize this material to build things such as surgical threads. So it's a very strong material, very flexible, and it is a natural material, so the body doesn't react to it. So here we have concept 5.2, carbohydrates serve as fuel and building material. This is not completely true. There are other functions. We can talk about that in class. Just remind me. But these carbohydrates are formed from monosaccharides that have multiples of CH2O. They can vary in their carbonyl position, producing aldose or ketose sugars, and they can vary in the number of carbons, right? So we get this full arrangement of different carbohydrates based upon the carbonyl position and number of carbons. They can also vary based upon the structural arrangement of the tetrahedrons in what we call chiral carbons, or carbons that have a tetrahedral nature to it. All right, these carbohydrates in a linear form can wrap around and form closed rings with an internal reaction which rearranges some of these atoms. This can form two positions, either an alpha position or a beta position for this hydroxyl. Disaccharides are formed when we take two of these monosaccharides and link them together with something called a glycosidic linkage. That is a dehydration reaction. And consequently, these are broken by a hydrolytic reaction, right? Which is the opposite of a dehydration reaction. Here we have maltose, which is a combination of glucose-glucose. Here we have sucrose, which is glucose and fructose. Polysaccharides are much longer chains, and they can have very large either storage or structural significance. Here we have starch from potatoes. Here we have glycogen from our cells. Very similar structures and built largely the same way, with alpha glucose linked together in a 1-to-4 linkage. Here we have cell walls, which are built differently. They are utilizing beta carbon in a 1-to-4 linkage, and this leads to the flip-flop nature and facilitates the formation of these hydrogen bonds, which gives you the ability to form these larger microfibrils, which can then go on to build the woody structures of plants or just simply the cell walls.